So food test for your starch, okay, 2 to 3 drops. Let me just highlight the key points. Okay, so I have got 2 to 3 drops of iodine solution. Okay, so if the solution remains the blue-black color, starch is present. Okay, so of course, if I were to put this enzyme called amylase into it, when we do digestion, you will see. Okay, so the purpose of amylase is to digest starch to glucose. So amylase digests starch to glucose. So once it is digested to glucose, it means that starch is no longer present. Okay, so since starch is no longer present, my solution will remain yellow-brown. Okay, because I am testing for starch, I'm not testing for glucose. Okay, but starch has been digested. So if I'm using Benedict solution, similarly I will have the observation yellow, red or green if digestion has taken place. Okay, so the moment the substances are digested and then it becomes a reducing sugar, it will turn up positive in a Benedict solution test. Okay, so I repeat, it will turn up positive in a Benedict solution test if there is amylase present to digest my starch into a reducing sugar. Okay, now the next one I have got proteins. So in proteins itself, if the protein is not digested, the solution will turn purple. Okay, it is not digested. Okay, protease, again same thing, ASE is the enzyme that will digest the proteins, okay, to amino acids. So let me write that down. So protease, okay, so once it is digested, there is no more proteins that are being present. So because there are no more proteins present, therefore it will not test positive for a burette test. Okay, Benedict test and burette test have very similar names, so find a way to memorize these two efficiently and separately. Okay. The last one we are looking at in terms of oil itself, okay, so that we will be using the emulsion test. So over here, the same thing, emulsion test, the purpose of it is to test for the fats. So if the lipase enzyme is present, and lipase itself, it will digest the fats to fatty acids and glycerol. Okay, so we will see the digestion component in the later part of it. So it will digest to fatty acids and glycerol. So once it has digested, there is no more fats, and because there are no more fats, therefore my ethanol emulsion test will not be positive. Okay, so the second part of this here, so it remains colorless, which means to say that the fats are digested if I have got lipase present. Okay, if the lipase is absent, no digestion takes place, so therefore you have got a white layer of emulsion.